Nakasagga. She's from Uganda in East Africa, and she's working with Dr. Setmari at Country Genetics and the corn breeding lab. She has a bachelor degree in agriculture science and entrepreneurship from Uganda Christian University, and she has a master degree in plant breeding and the seed system from Makerere University. She joined our lab 2018, and she has been working on perennial sorghum and the perennial maize. Thank you, Shakirah.
Howdy all, I'm Shakirah Nakasagga. I'm presenting my research on estimation of hydrogen cyanide, biomass yield, and an occupied area system-based regrowth in perennial sorghum and ZIA-derived lines. A brief background, sorghum is the fifth most important grain crop all over the world, and US is the world's leading producer with over 9.27 million metric tons. In the US, sorghum is the third most produced grain and is mainly utilized for forage and for biofuel, though there is an emerging small percentage that is being utilized for food since sorghum is gluten-free. Texas is the second largest producer of sorghum after Kansas. Sorghum has two species. We have the perennial species and the annual species. Under the perennial species, we have sorghum halpens, also known as Johnson grass, that is a tetraploid. We have sorghum propinquium, which is a diploid. We have sorghum almum, which is also a tetraploid. And these are all wild species. These are characterized by structures known as rhizomes. And rhizomes are horizontal underground stems that store nutrients and also are meristematic conduits for growth, meaning you can use them for vegetatively propagating sorghum. For the annual species, we have sorghum bicolor, and this is the domesticated species that is mostly grown for food around the other continents, like in Africa, in Asia. It's mainly utilized for food. So my research is being funded by the Land Institute in Kansas. The Land Institute is a non-profit organization that is mainly dealing in perennial agriculture. Several crops are being developed, perennial grains, perennial legumes, and also perennial oil crops. So for this research, the focus is on perennial sorghum, and the materials that I'm working with were developed by Dr. Stan Cox and Dr. Navkalu Fiona, with a long-term objective of producing a perennial grain sorghum. But so far, as they are within the breeding pipeline, right now we are assessing to see if they can also be used as a perennial forage or a bioenergy feedstock. Then why advocate for perennial? So far, previous research has shown that perennial sorghum has the overwintering capacity, and also it quickly regenerates from the rhizomes after unfavorable weather conditions. And also it has ecological benefits, whereby it increases the amount of the carbon print in the soil when it is cultivated. There are some limitations to the use of Johnson grass as a parent in these materials. As seen from previous research, Johnson grass has hydrogen cyanide toxicity if it's fed under certain weather condition to the ruminants. During extreme weathers of hot temperatures or very cold temperatures, it can produce hydrogen cyanide that is toxic to animals. And also, Johnson grass has been rated amongst the 10 most invasive weeds all over the globe. However, the materials that I'm working with, since it's a cross of Johnson grass with sorghum bicolor, which is the domesticated sorghum, that it is not invasive since the character or the trait is over 10% within the materials. And the other issue that that characterizes this material, getting a balance between the grain production and the vegetative yield is also kind of difficult, because not like naturally the Johnson grass is highly vegetative, like it produces so much vegetative mass, and therefore getting a way of allocating the resources from being used for vegetative production into grain yield also poses a limitation. However, we are doing selection to see how that can also be channeled. Also getting a cross between the Johnson grass and the sorghum bicolor, since there is movement from different ploidy levels, because the Johnson grass or the sorghum bicolor is a tetraploid and the annual species is a diploid. So crossability is kind of difficult, though we use colchicine in order to produce a tetraploid sorghum bicolor in order to increase the compatibility between the species. And also perennial breeding requires maintaining close year-round, which is kind of expensive or needs much more investment. And also the perennial evaluation is quite time-consuming, as I'll explain it further. So for the perennial evaluation, the classic method that is normally used is by counting the number of the rhizome-derived shoots, then taking the distance between the rhizome derived shoot and the crown, and also counting the number of the rhizome buds that are found underground. So the main objective of my research was to determine the hydrogen cyanide in a cross of sorghum bicolor with sorghum halpes derived lines, to estimate their biomass yield and composition, and also to use an occupied area system to predict earlier and more accurately which lines have the most early season regrowth and largest yield potential. For this objective, we want to see if we are to use the UAVs, will it reduce the time for evaluation for the perennial associated traits? And also an additional study is to differentiate structures that contribute to overwintering in Zea diploperennis, which is the perennial maize. So for the first study, the trial was set up here in College Station, and we evaluated a total of 100 materials, whereby the largest percentage was a cross of sorghum bicolor with sorghum halpens. Then we also added a cross of sorghum bicolor and sorghum propinquium, a pure line of sorghum halpens, and also grain sorghum. The population development of the largest percentage of this population, which is a cross of sorghum bicolor and sorghum halpens, was developed according to figure 3, whereby the sorghum bicolor was doubled using colchicine and then selfed, and the materials that I'm working with are the first back cross of the second filial generation. So these were planted in a randomized complete block design with 2 replications with 1 row plots. Hydrogen cyanide was collected last year in the late summer, then also this year in the mid-spring at regrowth, in the mid-summer at flowering stage, and also in the late summer at maturity. The protocol that was followed for the hydrogen cyanide evaluation was developed by the Texas A&M Veterinary Medical Diagnostic Laboratory, and the steps that I took for this analysis include, first of all, I sampled 3 to 5 plants in the field, got the fresh leaves, weighed 300 grams, and these were digested using chloroform and incubated at 37 degrees Celsius for 1 hour. So these are the flasks here before incubation. And then after incubation, the color change was scored from 0 to 4. This is the assay that is used at the Texas A&M Veterinary Medical Diagnostic Laboratory, which is a qualitative assay. So in order to make it more objective and get quantified values, I included calorimetry and got the L, A, and the B values. So the quantified color change was computed according to this equation, whereby L represents for lightness, A represents the green and red vector, and B represents the blue and the yellow vector. So in order to validate my results and also to make more definitive conclusions, I had to build a hydrogen cyanide curve, and this was built by varying the amount of amygdalin concentration. Amygdalin is the manufactured form of hydrogen cyanide, which is produced from bitter almonds. So I varied the amount of amygdalin solution from 0 to 0.03 grams, and this to this I added beta glucosidase, which is an enzyme that breaks down the amygdalin to produce the hydrogen cyanide. And using the qualitative assay, I used the indicator paper, and this is how the color changed from a very low concentration to a very high concentration. At a low concentration, the indicator paper stayed yellow, while at a very high concentration, it turned red. So the quantified color change, the values were processed in JMP. So this is the hydrogen cyanide curve, whereby this was the color change gotten from the equation, and this is the concentration of amygdalin concentration. And you can see from a very low concentration to the high concentration, there was increase in the color change. It turned more red. So for the results, for the first study, last year in the summer at maturity, there was no significant difference shown for the pedigree. And also when I made a comparison between the calorimetry and the qualitative assay for the color distribution for the different plots within the field, you can see most of the plots produced a low amount of hydrogen cyanide according to this color scale, whereby this one is for no hydrogen cyanide, 1 is for very low quantity of hydrogen cyanide, 2 is for low quantity of hydrogen cyanide, 3 is for a moderate amount of hydrogen cyanide, and 4 is for a very high amount of hydrogen cyanide. So this year in the mid-spring at regrowth, the results showed that the pedigrees were significant for the hydrogen cyanide that was produced. And you can see from the color distribution that most of them still produce a low amount of hydrogen cyanide, but now we were having also plenty of the plots producing moderate amount of hydrogen cyanide, and also we had some plots that produced very high amounts of hydrogen cyanide, and these shouldn't be given to the animals. So in the midsummer at flowering, there was no significant difference shown for the pedigree, and also this time the number of plots that did not produce hydrogen cyanide at all even increased further. And there were over 121 plots that did not produce hydrogen cyanide. We had only 3 that produced high amounts of hydrogen cyanide. So in the late summer this year, still the pedigree was not significant, but for the color distribution, more of the plots, they even increased further that did not produce hydrogen cyanide. And we had only 1 that produced high amounts of hydrogen cyanide. So when I combined all the 3 growth stages for this year, and grain and analysis of variance, stage was significant, but pedigree was not significant, meaning that, let's say at maturity, if most of the pedigrees, if they did not produce the hydrogen cyanide, then most of them were kind of similar and they were consistent, like they did not produce hydrogen cyanide. They either produced very low, and maybe at regrowth, if they the pedigrees also produced either very low, then maybe those that produced the high amounts or the moderate, they were also kind of consistent. Then when I graphed this, you can see that at regrowth, there was high amount of hydrogen cyanide that is in relation to the change in color. And this reduced the flowering and even further reduced at maturity. So from these study results, it can tell you that these materials produce more hydrogen cyanide at the earlier stages than at the later stages. Then when I combined, and when I combined data for last year and this year at the maturity stage, still there was no significant differences shown by the different pedigrees, but for the year was significant. And this can be explained that last year I got it at the time whereby the temperatures were higher than this year. So figure 9 shows a comparison of last year and this year at the maturity stage. And you can see clearly that in 2019, the amount of hydrogen cyanide was higher than for this year in 2020. So for the second study, it was mainly about estimating the biomass yield and composition. The biomass yield was obtained at a plot basis, and we got both the fresh and dry matter yields. And for the biomass composition, this was based on the dry matter yield, and we used near-infrared spectroscopy in order to analyze the biomass for the different components of cellulose, hemicellulose, lignin, crude protein, soluble carbohydrates, ash, and starch. So the results for the biomass yield, first of all, the pedigrees were significantly different for both the fresh and dry matter yield at 0.0001. And also, when I made a comparison between the grain sorghum and the perennial sorghum, results showed that the perennial sorghum had more than double of the yield in comparison to the grain sorghum, as you can see in figure 12 here. Then when a comparison was made between the perennial and the grain sorghum, following a selection of the highest yielding and the lowest yielding of the perennial sorghum to the grain sorghum, you can see that both the highest and even the lowest yielding had higher fresh matter and dry matter yield in comparison to the grain sorghum. For the biomass composition, the biomass had 30% of cellulose, 26% of cellulose, lignin was 13%, which showed that these materials have higher amounts of structural carbohydrates. But there was also a proportionate amount of the soluble carbohydrates, which was 12%. The ash was 8%, starch was 6%, and protein was 5%. And when an analysis of variance was run for the different biomass components, the pedigrees were significant for the cellulose, hemicellulose, and the starch. When a comparison was made between the perennial and the grain sorghum, the grain sorghum had significantly higher amount of ash and protein, while the perennial sorghum had significantly higher amount of soluble carbohydrates. When a PCA was run for the biomass composition, you can see here that the first two components explain most of the variation, And also you can see the grain sorghum. This is the one that I highlighted in the red boxes. It was spread out like throughout, like there was no clustering. So for the third study, it's mainly about evaluating regrowth and biomass yield using the UAVs. The UAV that we used is a rotary wing quadcopter with an RGB camera, and images were gotten for the sorghum trial for 2019 and also for the regrowth in 2020. And even up to now, images are still being taken. So the data from the images was processed, first of all, by mosaicing the images in ArgSoft meta shape. And I created shape files and cropped images in QGIS. And right now I'm analyzing in R in order to get the vegetation indices. That's the stage that I'm still on the third objective. And after that, I'll be making a regression between the vegetation indices and the classic characterization for the perennial associated traits. And this I will consider the number of the rhizome-derived shoots and also the number of the shoots from the crown. And also regression will be done between the vegetation indices and also the biomass yield. So for the fourth study, it is an additional study to this research, and it's mainly to do with perennial maize, and it's about characterizing of overwintering in Zea diploperennis. I have 5 genotypes, and these were planted in a randomized complete block design with 17 replications. The difference between the overwintering in perennial sorghum and in Zea diploperennis or perennial maize is that in perennial sorghum, so many studies have confirmed that the rhizome is what's responsible for the regrowth. However, in perennial maize, it has not been established since the regrowth comes from the crown and also comes from the rhizomes. So the question is, what could be contributing to the regrowth from the crown, since some of these lines, they can have regrowth but without rhizomes. So the steps that I took, first of all, was to check the composition of these different structures. A destructive sample was made whereby I got 2 plants per plot and separated the rhizomes from the crown. And these were oven dried, ground, and scanned using near-infrared spectroscopy. Then this generated a spectrum, which in combination with a rhizome curve, we made predictions, but the rhizome curve was developed by Washburn in 2013. And we got prediction of the different components that could be within the underground structures. So the analysis of variance revealed that pedigree was significant for the crude protein, the starch, the water-soluble carbohydrates, and the crude fat. And for the rhizome structures, they were significant for the crude protein, starch, and the crude fat. Figure 19 shows a comparison between the rhizome and the crown for these different components. And you can see that there is a general trend whereby the crown has more of all the assimilates than the rhizome. And this could give an insight as to why we also have regrowth from the crown, because composition is one of the factors that influences overwintering in perennial crops. So the conclusions from all the objectives, first of all, the first study, the perennial sorghum produces higher amount of hydrogen cyanide at earlier growth stages. Thus, fresh field should be given sparingly or ensiled at the earlier stages of regrowth. But then if it's to be given without being ensiled, maybe it would be preferable to give it at maybe flowering or at maturity. Then calorimetry is a potential tool for color quantification of the hydrogen cyanide qualitative assay and gives opportunity for data manipulation, increasing objectivity of the results. For the second study, the perennial sorghum is a potential source of forage and bioenergy, considering the high fresh and dry matter yields. And also the biomass had numerically higher structure and significant soluble carbohydrates, meaning that it can be used as a bioenergy feedstock, since these are some of the components that are looked at. Then the PCR revealed much variation for the biomass composition, which gives opportunity to make selection. However, the limitation to our study too was that we did not include forage and biomass sorghum in order to make comparisons. We used grain sorghum that really is not within the same range, like with the perennial sorghum. And for the third study, I'm still analyzing the data. I've not yet come up with any results, and also conclusions have not been made yet. Finally, for the fourth study, the underground structures were significant for the crude protein, starch, and crude fat, and these components are very essential for overwintering. But then there is need for further differentiation so that we can come up with definitive conclusions. And since all what is presented here, we still need to collect more data. And also the crown accumulates numerically more assimilates than the rhizome, and this could be a potential reason for overwintering and regrowth from the crown in perennial maize. But still, research is still ongoing. So the pending data that I'm still collecting, next week I'll be getting the biomass yield for this year, and I'll be processing soon for the biomass composition. Then the vegetation indices for 2019 and 2020 images will soon be obtained after running the associated analysis in R, and I'll be making a regression between the classic methodology and also the UAVs. And also we made our wet chemistry analysis, and then I'll be using the results to build a rhizome curve in order to better estimate underground structure composition using the materials that I have right now. And also there is still ongoing research to get the composition for this year for the crown and the rhizomes for the regrown maize. I'd like to take this opportunity to thank my committee, Dr. Seth Murray, my chair, Dr. Biruni, my co-chair, Dr. Russo Jessup, Dr. Soren Popescu, and Dr. Zujun Dong for their help and support in this research. I'd also like to thank Dr. Catherine Barr from the Texas Veterinary Medical Diagnostic Laboratory for helping me throughout the hydrogen cyanide assay, all the people at the quantitative maize breeding program, sorghum improvement program, and also I'd like to thank Mr. Bob Hodge and the Land Institute for the funding, Texas A&M University for the admission here to be a student, and also Texas A&M AgriLife Research for the resources used for this research. Thank you.
Great presentation. Thank you, Shakirah.
Right, thank you.
Do we have a question from a student? Okay, I'll ask one. Are you collecting seed for yield at this stage for your crosses, or only the biomass?
No, I'm not collecting seed for the yield. Right now, I'm mainly looking at the biomass.
I got a question. Can you hear me?
Yes, I can.
Okay, great. Are you using structure from motion to do that? I didn't see any software for structure from motion in your analysis.
No, I'm not using structure formation.
Oh, okay.
Yeah.
Because I guess I was wondering for biomass as a function of time and like with height, I was just wondering if that was being considered as an approach.
Okay, thank you.
Sure. A question I'm just going to put this out there. A question that I'm constantly asking people is, if we're monitoring perennial crops, how are we comparing them from year to year using these software systems? Because it's hard enough to make measurements. I've seen some different implementations from year to year.
Okay.
I guess if students are done asking questions, I had a couple related. Now, you made a cross between the two species, right?
Yes.
And I was wondering, what did you see as far as rhizomes in the progeny?
Okay. The rhizome in the progeny is not so vigorous like in the parent. Like if you are to make a comparison with sorghum halpens, also known as Johnson grass, the progeny does have the character or the trait, but it's like 10%, 12% if you are to compare it to the original wild parent. So
Yeah,
it's not invasive, yeah.
Is that across the board? You don't see segregation at all?
You see segregation. There are some that will produce like many rhizomes, and there are some that don't produce, and like in different ranges. But it's not invasive if you are to compare to the original parent.
And then one other question on those hybrids. We do some work with Johnson grass, and it's hard to germinate many of the seeds. Do you have some secret that you can say on how to germinate the seeds, or is, what percent do?
I've not had a very big problem with the germination of the seed for Johnson grass, because most of them do germinate.
Okay. Gypsum does, but the ones we get out of the wild don't seem to like to germinate at all. Thank you.
Okay.
Enjoyed your seminar.
Thank you.
And I missed the first few minutes of it, and you may have addressed this, but in your literature search, can you share with us, it should have come out of your literature search, can you share with us other crops and successes of converting an annual to a perennial?
Okay, I've read some papers in rice, because also they are developing perennial rice. And so far, the papers that I've read, there is some progress in getting a perennial rice crop.
But we don't know of any commercialized perennials that were perennialized from annuals.
Yeah, okay, yes, not yet there. Me too, I've not yet read anything on the commercialized perennials, but then the Land Institute is trying to produce these perennial crops, but then the problem is that for these perennial crops, if you are to compare with the annuals, their yield can never be comparable like in terms of grain yield, the perennial will always produce like less amount of grain, which maybe can be like a hindrance for it to be commercialized, especially with this industry that deals with the, you know, the hybrid that are very high producing. But the main aim or the objective is not to have like a very high or comparative perennial crop similar to the annual, because that's almost impossible. But to have a crop whereby it will always provide, even in terms, especially if you go to other continents, now like in Africa or in Asia, during periods of drought, you want a crop that will always provide something, even though it's not the best, but at least there is something other than not having at all.
I'm just going to make two brief corrections there, Shakirah. One thing you said is the perennial can never compete with the annual, but we don't actually know that because we haven't spent as much effort developing the perennial. So I think that's a very important word that maybe we need to be clear on, that we don't know it'll never compete. In fact, I think there's evidence it'll compete very well. But then the other one you didn't mention was Kernza, right? Kernza and the and pigeon pea are two that have been domesticated directly from perennials.
Okay.
But you're right. There's no annuals that have successfully been converted to perennials and are commercially available right now.
So could I get you to speculate then, from a Shakirah, from a evolutionary perspective, you get a perennial maize or a perennial sorghum. How long do you think it would be before it was commercially competitive with current annuals? What do you think is the evolutionary scale to move from developing a species, if you will, that doesn't exist in nature to making it commercially viable?
I cannot give a specific amount of years.
I hope not.
But there is still time and investment is needed to really come up with a perennial grain that is commercially available. There really needs to be some, because an example can be on the perennial maize. The materials that I'm working with, they were developed by Sheva, and he almost worked on them for over 35 years. And we are still trying to see how we can move this research forward. And also the perennial sorghum has not been developed today. There are some predecessors that, you know, worked with the sorghum before, but there've been some quite improvement, especially in the seed size. And maybe given like some few years to come, and if we also get technologies to shorten the evaluation time, the time frame will really reduce and we can have a commercialized perennial grain in the future.
So you're predicting that Dr. Murray will have that before he retires?
Probably.
No pressure, Seth, but you've got your work cut out for you now.
That's great. It gives me something to do. I don't like being bored.
Can I have a note about the perennial wheat that was developed by the Land Institute, Stan Cox and Lady Han and others? Actually, they crossed Triticum aestivum to Phaenoparam ponticum and Phaenoparam intermedium, and they have actually perennial wheat. And perennial wheat did really well in Washington State and Michigan. It didn't do well in Texas. We tried it everywhere in Texas. It would die in the summer. But perennial wheat is meant as a crop to grow in marginal areas. So it's not going to be grown where commercial crops are grown anyway. You need to seed it every 7 years. The seed is very poor, so you just need to target the biomass. But it has a very extensive root system So it's a very good way for carbon sequestration and actually occupying marginal lands. I have a question for Shakirah, which who gave really a very good presentation. This is a very good project, by the way.
Thank you.
Johnson grass is an invasive weed in almost all of the major all of the major field crops in Texas: corn, cotton, sorghum, and maize.
Yes.
So what's the potential of this hybrid becoming an invasive weed as well in the future if it gets adopted?
Okay, yes, Johnson grass is a very invasive weed, but the potential of these materials is that so far they were tested to see their invasiveness. And literature has it that 10 to 12%, these materials are 10 to 12% perennial, meaning that they cannot be invasive like the Johnson grass.
Okay.
Yes.
Thank you.
You're welcome.
So I have a question that's related to that. So if you want it to be non-invasive, but that also is going to decrease the perennial regrowth, right? So is there some kind of balance that you're trying to get with your crosses between lower number of rhizomes to decrease invasiveness, but enough to get good perennial regrowth?
Yeah, that's a very good question. Yes, like these materials, like okay, for example, you do not like collect, like you don't harvest like once in comparison to the annual sorghum, meaning that you can have at least maybe more harvests you can get of the grain, and it can kind of increase the yield these materials give. But then again, they are not that aggressive, like to be invasive. I don't know if I've explained that well. It's kind of tricky.
Thanks.
You're welcome.
Other questions for Shakirah?
Shakirah, did you look at the chromosomal level to look at perenniality? How many chromosomes are involved and all of that?
Yes, for these materials, they are tetraploids. The materials I'm working with are mainly tetraploids, the cross of the sorghum bicolor and the sorghum halpens. The sorghum bicolor had to be doubled because it's a diploid. So in order to increase the crossability and also the compatibility, it was treated with colchicine. And then the tetraploid sorghum bicolor was used to cross with the Johnson grass in order to get these materials.
Actually, my question pertains to the fact that you can bring in perenniality from Johnson grass without bringing all of the junk from Johnson grass. And to do that, you need to know the chromosomes that are impacted in perenniality per se. So you need to have enough perenniality without getting all of that genome, the Johnson grass genome. That's what I talked about. So do you have any information on what chromosomes, what genes are involved in the perenniality of
What I know,
hybrids?
okay,
Yeah.
what I know is that the perennial traits are like quantitative, it's a quantitative trait. So
Yeah.
it's controlled by many genes.
Yeah.
And this depends on the background that you are crossing with. Like in a tetraploid background, it's a quantitative inheritance. But if you go to the diploid background, if you are making a cross with sorghum propinquium, it is more like 1 to 2 genes. So it depends on the background you are working on.
Okay.
Yeah.
Thank you.
You're welcome.
So this is David Baltensberger, and I don't know, are you familiar with a Dr. Jungers at the University of Minnesota?
No, I'm not familiar with him.
Okay, he just received a $10 million CAP grant for developing and deploying a perennial grain crop enterprise to improve environmental quality and rural prosperity. He's at the University of Minnesota. They're working primarily with that wheatgrass stuff, I believe. But it's when Wayne said, "Is there a successful one?" That's the most successful one out there because it's generated $5 million in grants.
Okay, all right. Thank you.
Any last questions?
What about the yield? I think, Ellen, you asked about the yield. So does it produce seed? Does it produce enough seed?
Yes, it does
Okay.
produce seed, and you can be able to get at least one. Well, at least you can get more harvests, like more than harvesting it once. So that
how long does it take before you need to reseed? Does it take like 5 years or 7 years? What? How many years?
So far, like the ones that I've worked with, like they can be like still producing seed like up to 3 years.
Yeah.
But maybe more because the experiment was removed after 3 years, but they were still regrowing and producing seed.
Okay.
Yeah.
Thank you.
You're welcome.
I just wanted to compliment you on an outstanding seminar. Thank you for setting the pace.
Thank you so much.
Great job, Shakirah. Thanks.
Thank you. Thank you so much.
Well, if there are no more questions, thank you very much, Shakirah. Great presentation. I enjoyed hearing about it. And we will see you all again next week.
All right. Thank you.